My name's Levi Thompson, and as you probably know, I'm the Dean of the College of Engineering and a faculty member in Chemical and Biomolecular Engineering. And I have the honor to be your MC for today's lecture, which is a tremendous honor. I often tell people these are the kinds of events that, at least for me, are most enjoyable. To celebrate the excellence of our people, their performance, their contributions, their impact, especially having just we just had our convocation and commencement this weekend, and sort of an extension of that, you know, to celebrate the excellence of one of our faculty members. And so formally, I want to welcome you to this named professor lectureship. And I'm going to not only welcome those of you who are here in the FinTech Hub, but those of you who are joining us online. You know, I mentioned that this is a tremendously rewarding thing for me, and it also is for the recipient of the named professorship. And today we're here to recognize Darrin Pochan.
Perfect.
Okay.
Pochan.
I practiced that for 5 years. As the Distinguished Professor of Material Science. This is an endowed professorship that is one of the most prized professorships in our college. The criteria for becoming a named professor, they're both simple and difficult at the same time. First, you have to be viewed as among the top 10% in your research discipline as determined by your peers. Second, you're expected to be an effective teacher and to have made significant contributions to your department's educational mission. Finally, you're expected to have served and contributed effectively to your department. By every measure, Darrin has exceeded these requirements. Here we are in the midst of sort of merit review. Darrin has certainly exceeded all of the requirements. Darrin joined the University of Delaware in the MSc Department of Material Science and Engineering department in 1999 after earning his PhD in Polymer Science and Engineering from the University of Massachusetts, Amherst, and a National Research Council postdoctoral fellowship at the National Institute of Standards and Technology. Darrin's research focuses on the construction of new materials and nanostructures via molecular solution assembly mechanisms. Darrin's scholarship has been described in more than 160 papers. I think it's 164, 65?
Give or take.
Give or take, okay. But more than 160 papers with nearly 26,000 citations. And you can do the math there. That would lead to an H-index of 84, which is extraordinary. He is a co-inventor on 6 patents and has given more than 235 presentations, including invited lectures and keynote lectures. Not surprising that Darrin has been recognized with a number of prominent national and international awards, including the John H. Dillon Medal from the American Physical Society, which is amongst the highest honors attainable in his field. He's also an elected fellow of the American Institute of Medical and Biological Engineering, the American Chemical Society Polymer Material Science and Engineering Division, the American Physical Society, and the Royal Society of Chemistry. His dedication has led to a reputation as both a leader and a mentor. I imagine that some of you here probably, I think we all may fit into that one of those categories. Certainly, I have found that Darrin is a tremendous role model, especially during the time that he served as department chair. He served the department for 8 years in that capacity. During that time, the department grew its faculty, its scholarship, its graduate programs. They even started a new undergraduate degree program. You know, for those of you who have been in programs that have existed for years and years, just imagine what the start of that program may have looked like. And Darrin under Darrin's leadership, we now have an MC department with a undergraduate program and graduates. We had graduates this past weekend and graduates, I think, last year. Was that your first year?
Second class this year, yeah.
Second class this year. So just tremendous contributions to our college and to the University of Delaware. He and his colleagues successfully led and brought to the University of Delaware's campus the first ever National Science Foundation Materials Research Science and Engineering Center, or MERSEC, the Center for Hybrid, Active, and Reactive Materials, or CHARM, placing the MSc department here at the University of Delaware among the very elite material science departments in this country. In fact, at the time I came to the University of Delaware from the University of Michigan, Michigan didn't have a MERSEC. They have one now, but Michigan didn't have a MERSEC, and the University of Delaware did, so very proud of that. He has been PI or co-PI on more than $40 million in research funding from a variety of different agencies, too many to name, including receiving an NSF Career Award. As an educator, he's trained 22 PhD students, 2 master's degree students, and 5 postdoctoral scholars here at the University of Delaware. He's already up. Darrin, please join me at the podium. And recognition. In recognition of your outstanding performance, we have a congratulatory letter
Thank you very much.
and a symbolic medal that we hope you will wear, seriously, we hope you will wear during ceremonial events, including commencement and convocation. So, you know,
Right on. Thank you very much.
how's that?
It's heavy.
So on behalf of the College of Engineering and the University of Delaware, it's my pleasure to present you Darrin Pochan, the Distinguished Professor of Material Science. And very much look forward to your talk entitled Building Materials at the Nanoscale for Molecular Self-Assembly. Seeing is believing. Darrin?
Thanks, Levi. Thank you very much for that introduction, and I'm ecstatic to have all of you here today. It's a huge honor for me to get this named professorship and to be part of a crew of people who I've enjoyed working with and I've looked up to since I've been here at the University of Delaware. So I very much look forward to interacting with that group. And I'm going to talk a lot about that during my talk today with respect to the people with whom I want to continue interacting with for the rest of my career here at the University of Delaware. The first slide needs, one big fat cliche here to a general scientific audience, but it points out something very specific I'm going to focus on, and that is I'm going to focus on my friend, the transmission electron microscope. So I'm going to talk some about things we do for research in my research group, and also almost all of that work is collaborative with other research groups on campus and outside of the University of Delaware. But in particular, I do want you to take home the point that a lot of times I think real space, microscopic characterization, looking at things, seeing an image, a lot of times that gets belittled in the scientific literature, and I'm here to say that seeing is believing, and images are really important with modern material science research and lots of other fields also. We can talk more about that during the talk and afterward if you're interested. But we're going to do some exotic things. I'm going to talk about some exotic nanoparticles that you don't normally see. But maybe most importantly, a third title slide. And that is, none of this happens without a bunch of other people working with me, faculty, colleague level, graduate student, undergraduate student level, to get any of this stuff done. So that's something I want to talk a little bit about first with respect to having success in research. Very quick history lesson. I stole some slides from my dean interview process here to highlight the history of the department, to highlight a couple things about people. And that is, Levi was mentioning the first undergraduate classes in the department ever this last 2 years. But the department is only 25 years old this year, 26 years old this year. It wasn't officially even started in 1996 when one person was hired, John Raybolt from IBM, to come be the founding chairperson of the department. But he spent a couple of years recruiting, and I don't know if anybody knows, this is a shout out to John. I'm just going to leave it at that. If you're interested in what that means, I can tell you about it after the talk, that yellow arrow. 4 core faculty members hired from industry, so a really unique core to the department. Everybody from industry. John from IBM, Ismat from DuPont, Jingwang Chen from Exxon, Mary Galvin from Bell Labs. And so then those very accomplished people from industry were crazy enough to hire the first academic person, which was me, as the first purely academic assistant professor in the department. But I show this slide to bring up another important point. That is, the department was really small back then, right? So this wasn't big enough for a legit search committee. So during my interview process, there was another member, and that's Doug Doran, who a lot of you know, a great guy, chemistry professor, computational chemist. He actually interacted with the department a lot, research-wise over the years, doing computational chemistry and inorganic materials. This is what he looks like happy, though. But if you've ever seen him during a talk, like if you're staying up here seeing him in the audience, very Ron Swanson, like just sitting there staring at you like, what is that guy talking about? So during my interview, I was just like, well, this is a good exercise. This is good practice. I'll hopefully get some offers other places, because this guy is not a fan. But then I talked to him after, and he was like, oh, that was great. This is really exciting stuff. And I was like, wow. Dude, look in a mirror when you're at talks, man. That's rough. So that was back in 1999, but this is today. And so this is the Material Science and Engineering Department faculty. Lots of co-appointments across the college and also across the university, which I think really highlights the collegial, collaborative nature of the place. And this group of people, I'm just so proud and happy to be working with this group. And this is one of the main reasons that I'm here at the University of Delaware. Sadly, since we're a new department, we have our first ever retirements. I say this to other people in other departments. They're like, whatever, man. Like, we have to deal with this all the time. People are always leaving and retiring. But Michael, Bob, John, and Chow, just gigantic gaping holes in the culture of the department with these guys moving on and retiring from their active faculty positions. But as an entire group, just a fantastic group of people to work with on a daily and a weekly basis. But I highlight this slide to point out a couple of facts about my publishing record that Levi mentioned briefly during the introduction. Out of all of my papers to date, and certainly going forward, there's only 8 papers that have just Pochan and students, or Pochan and postdoc or postdocs on the paper. 150 plus papers, and all the papers going forward all have at least 1 co-PI, if not 2 co-PIs or more. And they're shown up here. So Xin Chao, Arthi, Ismat, Christy, Thomas, Chris, Michael, Chow, Mary, let me stick here, Eric, Chris, and Norm. These are people still at the, mostly still at the University of Delaware. Mary's not, she's still here, but still actively collaborating with a lot of these people. Very fun people to work with, very smart people to work with. These are people that used to be at UD who have left now. This is Cindy Carson, Randy Duncan, Sylvain Cloutier, Eric Kaler, Eric Block, and Joel Schneider. I'll talk more about Joel Schneider later, but also great people to have worked with. And here's people currently at the University of Delaware: Charles, Lore, April, Wilfred, Tom, and LaShonda. We have papers and collaborations in various points of progress, papers forthcoming. This is what gets me excited about doing research, doing stuff with other people, synthesizing ideas with other people with other fields. That gets me really jazzed up. That's been core to my whole career, and I look forward to taking that forward. Also, Jeff Savin and Karen Woolley, I'll talk a lot about these two important collaborators outside of the University of Delaware that have been really important to my career. So collaboration, just absolutely key and core to just everything that I've been able to do here at the university. But just as, or maybe more importantly, are the students that do all of this research in the laboratory. This is not unique to me. Any PI, I think, would say this over and over again. This is actually a photograph from my first cohort of students that existed in my group before I got tenure. So they nicely put together a picture and officially called it Darrin Pochan's tenure package. It's just absolutely true. But Honggung, Rohan, Matt Lam, he's currently on our advisory board. So those of you from MSc recognize Matt, who's now at Merck. Vahik, Balent, Nikhil, Kelly, Tuna, and Lisa, and Jabin, a postdoc. That group, just a fantastic core. You all know when you all got tenure, like you get tenure because of these people. You can have all the greatest ideas in the world, but if the students aren't succeeding, it just doesn't happen. And they were just a fantastic group. I didn't mention these two. If you want to talk about them later, we can talk about them later. I don't want to talk about them too much. So that launches an academic career, and then a bunch of other people you get to work with then throughout your career when you start to get older, like myself. People like Kyung-hee, Ji, Nariti, and more recently, Yi, Haoja, Zihan, Yao, James, Tianren, Bree, Jacob, Weiran, Matt, Amanda. This was just this last Christmas celebrating at Grottos. Couple more people. Jessie. Jessie Soon's still pulling her weight on the MSc homepage. So she's working every day for us, bringing new people to the university. So Jessie, Aisha Gul, Samir, Shang, Jiahua, Pete, Peter, Frank, that's Frank, not Peter. Frank, Kenny, you, Beth, Tsonsi, Mike Hayter, so busy I could only find pictures of him in the hutch at the synchrotron. Didn't find any pictures of him just hanging out. And then more recently, Wenjun, Bin, and Siri. I wanted to go through all those people individually. Just fantastic group of people to work with, still working with a lot of them going forward. One thing I don't have time to do is to put all the students I've worked with in other people's research groups. So the collaborating students, I just consider us one big group to work together to do all of our really fun research together. So I'm gonna sing the accolades of these people throughout the talk this afternoon. Okay, but we're gonna keep it really simple with respect to molecules. We're gonna design molecules. We're gonna put them in a solution, dissolve them in a solvent, but they're gonna have some kind of interaction together where they're gonna zip up and build our exotic nanoparticles. And that's where the microscopy and looking at the structure is gonna become really key to understanding what we've made. But these are the three I'm gonna talk about. I'm gonna start with some peptides. Then I'm gonna go to some blockopolymers. So if you're not familiar with what these things are, I'll tell you what these things are. But then, because I like them so much, we're gonna come back to some peptides at the end. And the inspiration for all this is really bio-inspired, naturally inspired, because nature, and particularly with peptides and proteins, nature does it best. And so all of our favorite topic recently, viruses. Beautiful nanostructures, 100 nanometer scale bars in all of these images. 3 different kinds of viruses. This is an adenovirus here, where you can see these spikes, and you see this really clear cage-like structure, and you can see these little bumps on the cage. These are individual proteins that when they're in solution, they interact with each other and build just this very specific structure. This gives you a cold, a common cold, so you get these all the time. Don't touch your eyes or your face, then you won't get a cold as often, but these get on your hands. They're stable for like weeks, for months. They can dry out. They're crazy stable. Once they get in your nose or in your eyes, you get a cold. This one's maybe everybody's favorite. This is SARS-CoV-2 coronavirus. Similar to this, but you see it's got a little stable particle, and it's got this crown-like display of things sticking out the end that helps it interact with your cells and get you very sick very fast. This one you don't ever want to catch. It's very characteristic looking, and it's an Ebola virus, but to point out the fact that they don't have to be these round little cages, they can be these really ornate, longer structures. So these are all things that make us sick, but nature really has some of the best examples of viruses that attack other things, like bacteria or arachne, in nature. So these are all viruses that attack bacteria. Some of the most beautiful assemblies are these filamentous assemblies. This is a 300 nanometer scale bar, or excuse me, 100 nanometers here, scale bar showing you the length scale of these things. Individual proteins zip up into these really long columns, wrapping around, in this case, RNA. So when it attaches to a bacteria, it can deliver the RNA and impact the machinery of the bacterial cell. But they can just be the most beautiful structures. So maybe you've seen these in the literature. This is a bacteriophage, War of the Worlds nanorobot that attaches the bacteria and squirts its payload into the bacteria. Just horrifying, beautiful structure, horrifying mechanism. Can you assemble this synthetically yet? No, but this is what inspires us to be able to make new assemblies. But this is maybe my favorite that I learned at a seminar several years ago from a virologist at Montana State. So Montana State, smaller school, very famous virology department because it's right beside Yellowstone National Park. And within the hot springs of Yellowstone National Park, there's really unique organisms that live just there, and they have their own unique viruses that live right there to interact and affect those organisms. And they look just like champagne bottles. So somebody's got a sense of humor. That's my favorite virus on the face of the earth. But all of these beautiful, all of them nanoscopic. So all of these have a 100 nanometer scale bar. And if you're not constantly dealing on the nanoscale like lots of us do, 100 nanometers is about 1/500th of a human hair. Human hair is going to come up, ironically, a lot during this talk. But that's the length scale of what 100 nanometers is. But nothing's safe. These have evolved over billions of years to interact with all of the living organisms on Earth. So these kind of structures inspire us. Can we design molecules to build these kinds of nanostructures and have these kinds of really deep function synthetically and in the laboratory? So that's what self-assembly is all about. All right, let's start simple. This is our first example. We're going to build some one-dimensional things. And let me just dive in and show you exactly what the molecules look like. This was a close collaboration with Joel Schneider. I'm going to talk more about in a minute. He and I met during new faculty orientation, like when you learn about your benefits and stuff. We were just kind of hanging out. And I was thinking, oh, you're in the chemistry department, I'm in material science. So we just chatted. And then we were at a dinner several days later for new faculty, and we talked a little bit more in depth. And he said, oh, I work on peptide design. I said, oh, I work on soft materials self-assembly. And so we kicked off a collaboration within like 72 hours of starting work. So that was a pretty quick clip and indicative, I think, of things to come at UD. But the molecule shown here in the folded state has a really unique conformation. It looks like a hairpin. So it's called a beta hairpin because it looks like a hairpin when it's folded. And it's also stabilized by a lot of beta sheet hydrogen bonding. So what's the big deal about that? The big deal is if you have solution conditions where this hydrogen bonding can't form, low pH, a lot of salt, and also high temperature, you can have the molecule freely soluble in solution and unstructured. But if you add a bunch of salt, for example, and screen electrostatic interactions between amino acids on the arms, the turn sequence forces the arms to fold into this beta hairpin. So why is that unique? That folding then predicates a self-assembly event, because once it folds, all of the hydrophobic chemistry is on one side, all the hydrophilic chemistry is on the other side, and then in the plane of the folded molecule, you can have additional hydrogen bonds form. So basically what you have is a polymerization event that happens, but only after the molecule folds. So you have a trigger. If you control this process, you dictate this process, and you get self-assembly into a one-dimensional like polymer that in fact make these really unique hydrogels, which I'll talk about for a little bit in a slide. But I really want to highlight the person who designed these molecules, with whom I've worked with for decades at this point. This is him. This is Joel Schneider. First of all, I did not stage this photograph. This whole photograph looks like a caption contest, right? So it's like, okay, what's going on here? This is his postdoc advisor, Bill DeGrotto. I don't know what he's doing back there, but he's just like talking to himself. But this was at the ACS meeting this spring, where Joel won a really big award from the ACS for like a Lifetime Achievement Award in the Biological Sciences Division. Here he's second guessing this one for the road, because we just had lots of Sazeracs in New Orleans that whole night. But this is Joel. He knows how to celebrate also, in addition to coming up with some really great ideas for research. But The thing I want to point out is this was just all cartoon, right, and hypothesis. And our initial electron microscopy taken by Balint for his thesis did in fact prove that we were making this nanofibrillar structure. And so this image over here, 100 nanometer length scale bar here, with this small nanoscale plate of spaghetti, proves that we are getting this nanofibrillar structure. The dimensions are the same as the approximate molecular dimensions. This image was really hard to get because it turns out these molecules make these stiff gels. So how do you get a stiff solid into a thin enough grid to put in the electron microscope and characterize it? It was really challenging. Balint spent a lot of time figuring out how to do that. So the sizes do match. So that was our first proof, microscopic proof that we did make what we designed to make. But just to highlight the fact that that assembly event, these one-dimensional assemblies make these really interesting hydrogel materials, physical hydrogel materials that we study to this date. And Joel Schneider also studies to the date. And one of the things that makes them interesting is, if you look here on the right, this is about a milliliter of gel that's assembled around a living cell population. So the point is, it's a physical assembly of peptides. It's very biocompatible, so you can encapsulate things like living cells, you can encapsulate drugs, you can encapsulate proteins, you can do all kinds of encapsulations to then do fundamental biological sciences. And another interesting property is you can assemble 3 layers of the gel, and because it's a physical gel, if you put enough force on that material, in fact, if you go above its yield stress, it will flow like a liquid, and then when you cease the shear, it immediately snaps back into a solid material. And so this allows you to do things like the following. You can make a layer of gel, make 2 layers of gel, and then you can make channels of a 3rd gel in the 2 layers. Turns out this actually recapitulates the outer cerebellum. So this is some materials that I'm working on with Sigrid Longhans at Nemours Hospital to try and mimic the healthy development of neuronal cells and also disease states in the brain. So this morphological control is important with a shear thinning gel like this. It's also important for the idea of delivering drugs. You want to deliver a gel in vivo or then can elute drugs at, like, say, a cancer site. So a really rich a behavior that provides a richness in applications of these materials, both fundamental and also therapeutic. But it was an important puzzle that microscopy helped us out. And so what you're seeing here is small angle neutron scattering data. The point is we went down to NIST. Lots of people in the room like to do this too. We go down to NIST where there's a nuclear reactor and you can use neutrons coming off the nuclear reactor to characterize materials. And so you put your gel, the neutrons scatter off your material, and you can look at the scattering pattern and figure out what your nanoscale structure is on a global scale. You can look at your entire sample as opposed to a microscope where you're looking very locally in your sample. So they're very complementary techniques. But importantly here, intensity versus scattering wave vector or scattering angle, the important thing I want to show you is all these curves look the same, right? There's 3 different gels, slightly different solution conditions, and this -1 slope here, this -1 scaling tells you you've got some one-dimensional structure. I already showed you the microscope image. We knew we had one-dimensional structures, but we have this stiff gel. So the interesting thing is if you also put any other one-dimensional structure in the beam, carbon nanotube, inorganic nanorod, worm-like micelle, they all give you almost identical scattering. Those things aren't cross-linked in any kind of network. So what gives us our hydrogel properties? Because the scattering looks just like any other nanorod structure, that's where the TEM came to the rescue again. So this was Tuna Ugel's thesis, where now he did TEM on networks made from the self-assembly of the peptides, but he did cryo-TEM, where there's still solvent around. So the light gray background is actually buffer solution, and the light gray squiggles you see are the actual nanofibers trapped in the cryogenically frozen vitrified hydrogel network. And so what you'll see here at the blue arrow, well, you look at the red arrows first. The red arrows are where 2 fibrils cross over each other and form an entanglement, which is one form of a cross-link to give you a physical stiffness in a hydrogel network. But importantly, the blue arrows point to where a single fibril branches off into 2 daughter fibrils of the same size. And these branch points, this is the best one. Where's the best one? This is the best one right there. These branch points are another form of crosslink that we couldn't have discerned otherwise without seeing it. Those branch points, we hypothesize, are like this, with this hydrophobic core of the peptides. They don't have to be perfectly assembled like that. They can come down off axis, and then you have a defect from which a new fibril can form from the existing parent fibril. Wouldn't have known any of that mechanism without seeing it in the microscope. So cryo-TEM came to the rescue and really showed us exactly what was going on at the nanoscale. And heroic imaging effort by Tuna Ugel back with older equipment, newer technique to get these kind of images. Okay, one other thing with the gels, highlighting Tuna's work, is that he then got really interested in correlating what the nanostructure was with the physical properties of the networks at different concentrations. And so he did things like looked at different concentrations, from low viscosity liquids to stiff gels. What did the nanostructure look like between those three things? And he saw the following: when it's a low viscosity solution, the peptides are just starting to assemble, they make these little clusters. So we didn't know if they made individual fibers everywhere isotropically that eventually start to interact. They don't do that. They nucleate and grow in these small clusters. These clusters start growing in size. When they start growing in size, the fibrils start getting longer. Now the viscosity starts to get a lot higher because you've got these long chains in solution. Then eventually, here's a cluster here, here's a cluster here. They start to interpenetrate, and immediately it percolates and turns into a stiff solid. So with microscopy, Tuna was able to see the nanostructure directly relate to the macroscopic properties of the solution versus the really stiff gel. Okay, I'm going to end this initial part talking about the gels with a little foray into a failed business venture. And that is, a lot of us are engineers in the audience. We talk about entrepreneurship all the time. We're doing fundamental research. We want to see what the funding agencies want to say, hey, what's that going to relate to as far as potential technology? If you can make that connection, you're like, hey, we could start a company. We could do this ourselves. We're going to get rich. It's going to be amazing. The small group that started this work formed a company because we recognized this idea of being able to have a shear thinning gel that is biocompatible, cytocompatible, deliver drugs. We were really excited. So we started this company, and then very quickly, a couple years later, dissolved that company because we would go out and talk to other companies, and they were like, you need to do so much more work before we're interested in this. You've got what, a couple papers? And they patted us on our head, metaphorically, and sent us on our way. It was a really good experience to have that experience, to know what it takes to get a company interested, to know the amount of work it takes, particularly in biomedical things, to have things become a technology and not just an interesting publication. And so it was really gratifying just this, at the end of this last year, we did patent it. The patent was licensed by Manav Mehta, who started a company called Gel for Med. He was looking at a bunch of different gel technologies for wound healing, topical wound healing applications, and he got it through FDA approval just recently. So he licensed it, I don't know if you guys know off the top of your head, he licensed it like 12 years ago?
2018.
All right, 2018. So that's not 12 years ago. Maybe we started talking 12 years ago, I don't remember, but in 2018, and then 6 years later with him working every day, it finally got FDA approved. So again, to my naivete, when we were going to start the company and do everything ourselves, it took a whole separate company to take this through FDA approval to now start to use. But it's just super gratifying to see 20 years on that the material is of interest and now is going to be used in a therapy. Okay, big switch to a couple different other molecules here before the end of the talk. And that is, I want to first focus on the collaborators. The people shown, these are all people I've published with outside of UD. Had the opportunity to work with just really great people all over the place, mostly meeting them at conferences. I met a lot of these people through Soft Matter, the journal I was on the editorial board for many, many years. I learned so much from these people and trying to apply it to my own work. But I highlight some people who are particularly important. This is Karen Woolley, showing her in her natural environment while she's collaborating with people. She's just like the most energetic, productive person I know. I don't know how one does that. She's quite unique in that sense. So I'm going to talk about her molecules in a minute. Current collaboration with Sigrid Longhans at Nemours Hospital to do this neuronal neuronal biology study. Tim Deming, I don't have time to talk about Tim, but he was one of the first collaborators I had at UD looking at block copolypeptides. Ishi Tallman and particularly Ganit Danino, her in particular taught us all about cryo-TEM. They were one of the first, he was one of the first people to use cryo-TEM outside of biology. And then people saw the possibilities, and I think the rest is history because now it's ubiquitous everywhere. And also Jeff Saban, shown here with his wife Phoebe. Turns out Jeff, Beth and I have been friends with Jeff and Phoebe for many, many years outside of work because Jeff's at Penn. He was friends with one of Beth's friends from her postdoc in Japan. Go figure. So we were hanging out with them when we lived in Philadelphia 20 years ago. But then we started to work together more recently. So I'm going to talk a lot more about Jeff. Okay, so a brief foray into block copolymers from the Karen Woolley group. Very different molecules, not amino acids like proteins. Now we're talking synthetic polymers, but synthetic polymers where you have one kind of chemistry connected to another kind of chemistry in one chain, usually amphiphilic. So one part likes water, one part doesn't like water. And really well known in the amphiphilic idea is that if you change the ratio of these two, you can go between a flat membranous assembly, a more cylindrical rod-like assembly, or a little spherical assembly. So change the molecule. You can have the same chemistry, just change the ratio of the two, put them into water, the hydrophobic groups come together, and you can have different curvatures that give you these different shapes, essentially on the nanoscale. So we wanted to do something different. So this is the crux of what we did. I'm just showing you kind of we'll talk a little bit more in detail about each of the, some of these going forward, but we wanted to divorce the assembly from the molecule. And so we used kinetic pathway to do that, and we also used complexation to do that. And so we made cylinders where the chemistry is not a core shell chemistry, but it alternates in chemistry perpendicular to the cylinder. Also phase separation within the cylinder to give it an undulation and curvature. Nano donuts, where they're locally cylinders, but they all close off into toroidal donut-like assemblies. Telephone cord assembly, helical assembly of a cylinder, and then also stacks of poker chips. So these are individual discs that stack together, but when you dry them out, it's like you push the stack over. You did it really well at Vegas and pushed them over at the end of the night. How do we form these things? So this is, again, a really close collaboration over the years with the Woolley lab. They are very good at making all kinds of different flavors of block copolymers. So these are some pretty simple ones. Shown here on the left, there's two hydrophobic blocks connected together, a styrene and methyl acrylate, if you're interested in the chemistry. And then importantly, the hydrophilic block that likes water is acrylic acid, really common in lots of industrial polymers, but importantly in water, weakly acidic, so it's slightly negatively charged. So when you put this in solution at these ratios, it makes spheres. So what we did to change that was we threw in this case, different diamines, a short diamine, a longer diamine, and an even longer diamine. The amine complexes with the acid and forces the amine molecule to hang out in the hydrophilic group. So that does two things. One is when it complexes with the acids, this amine acid complexation is really strong. When it complexes with the acids, it pulls the acrylic acid chains together and also makes them collapse. But then it also fills this region with volume. So you can control the volume of the hydrophilic phase separate from the length of the hydrophilic block. And so, for example, at the same ratio of amine to acid, but with these three different diamines, you can have spheres, cylinders, or discs in this case, all with the same block copolymer. So it completely divorces the nanostructure from the block copolymer composition. So what other games have we done over the years with this? One important thing you can do with this complexation is you can trap two different polymers together into the same particle that otherwise thermodynamically don't want to hang out with each other. So fundamentally, all the stuff I'm showing you now, not even close to equilibrium. All these things are kinetically trapped, but reproducible and stable assemblies. And in this case, two block copolymers, but with different hydrophobic blocks, styrene and perfluorostyrene. These two things want to phase separate from one another. What happens if you take these two polymers, add amines that interact with their acrylic acid groups, force them to be complex together, and then force them into the same micelle? You can force these two polymers, these two different core blocks, into the core of one cylinder. And when you do that, they phase separate but just locally, just within the core of the micelle. You have the fluorinated block go to one domain, and the non-fluorinated block go to another domain. And so then you get these wiggly cylinders. They're not unstable. It's not a Rayleigh instability where they're breaking up into droplets, which is a common thing to think when you see this. They're stable over time because of the nanophase separation within the core of the cylinder. Okay, a final couple of other images of fun exotic micelles you can make. If you do this complexation game, you can make cylinders. The complex of the amines with the acid also makes the shell of the particle attractive to itself. And so if it's making a cylinder, the ends are a defect. So the system wants to get rid of the ends. So how does it get rid of the ends? One of the mechanisms is it closes off on itself and makes nano Cheerios. This is a cast film stain. This is in cryo-TEM, just showing they're stable in two different ways. But this one is maybe one of the most fun ones because this one highlights serendipity in the lab and mistakes in the lab leading to good things. And so this is the same kind of block of polymer, now with a more complicated multi-amine. Now there's 4 amine groups in this molecule. But importantly, this is Sheng Zhang's thesis. He came to me for many months looking at the assembly of these two, the polymer and the amine, and his job was to make a 1-to-1 amine to acid ratio. Right? So add enough of the multi-amine with the block of polymer in a solution so that every amine can find one acid group to complex. And he came for months showing me TEM images of nothing, like blank ones. And I was like, Sheng, if you bring another blank TEM image in this office, I don't need to see another blank image. He's like, but I don't know where anything is. And I said, well, polymers are really big molecular weight. They don't evaporate. So they're in there somewhere. I don't know what to tell you. And then we were in the lab and he showed me a vial one day, and the vial was crystal clear, but it had these big white things floating around, looked like dust. I was like, you gotta look at those because that's the only thing left in your solution. It turns out that was the assembly. So they were like hundreds of microns in length. Just there was like one particle in the solution. So that's why he wasn't seeing anything, because there was like one particle to find. And it's making, what you're seeing here is a projection of like a old-fashioned telephone cord. So the complexation of the amines into the shell of the cylinder caused collapse and stress in the shell of the cylinder that caused it to go through an instability and buckle up into a really perfect helical structure. He could control the pitch of the helix, and sometimes there were double helices, sometimes there were triple helices. It was really cool, but completely by accident. Also, I forgot the accident part. He was also looking at 10-to-1 amino acid ratio. If you go 1-to-1, it all goes away and it makes cylinders, spheres, and disks depending on the ratio. So the accident of doing the calculation wrong off by an order of magnitude, he got these really cool structures. So don't throw away those solutions that are the wrong, Kenny. Don't throw them away. Make sure you look at them because there might be something really good in there. All right, now final couple of examples of trapping different molecules together is you can make some really interesting hybrid nanostructures. Polymers that make vesicles, polymers that make cylinders, you trap them together, they'll make a hybrid. Part of it will be a vesicle, like a flat membrane, and then part of it will be a cylinder. So you can make little nano hockey clubs and hockey pucks, hockey sticks. You can also take vesicles, mix them with spheres, and I don't know what you want to call these, but you can get little membrane sections with little nodes of spheres stabilizing them around the edges. We started uncovering just some really weird assemblies that were really fun to report. We don't really understand necessarily exactly why these are stable still, and we don't work in these anymore, so it's out there in the literature if anybody wants to take a stab at really explaining how these things are forming. Let me skip past this for time and show you one more example of locopolymers. This nanostructure is formed from a simple dye block, hydrophobic block, hydrophilic block. This is cryo-TEM, 500 nanometers, showing these vesicles. So these are membranes that wrap around, they're spheres, they have solvent on the inside, solvent on the outside. This is a really common vector to deliver drugs in vivo. People make these kinds of nanoparticles and they encapsulate drugs on the inside, put things on the outside to make them compatible with various parts of your body, and then they put them in your body and try to deliver those compounds different places. Quick example of how microscopy is really key in showing modern technologies working. So this is a collaboration with Mike Mitchell at Penn. we're just doing some imaging to look at their nanostructures, but what they're making are RNA, so this is similar technology to vaccines, RNA delivery to inside the body, but with vesicles. And so they want to make vesicles that have RNA complex on the inside. Here's a formulation that worked. So this is the cryo-TEM. You can see the vesicles. They've got like a complicated pattern on the inside because there's a lot of RNA on the inside with other molecules. But importantly, here's a formulation that didn't work. You can see the RNA-rich ones, but then you can see these other ones with these weird vesicles kind of sticking, Frankenstein vesicles sticking off the ends. The formulation process for this one versus the first one, almost identical, just one slight change. This would not pass muster with respect to a delivery vehicle. You want them much more homogeneous. So again, the microscopy comes to the rescue to show exactly the difference between the two formulations. Okay, and to end, a handful of slides about some new stuff, newer stuff. This is a close collaboration with the Savant Group. It was a collaboration started with Christy Kick. It's an ongoing collaboration also with Chris Clarkson, and to emphasize, a bunch of other people now through the MRSEC. So I don't want to sound exclusive. This is a really fun group of people to work with on these kind of fundamental building blocks. And this is really an idea that comes out of the Savant Group. These are called coiled coils. There's 4 peptides here, 4 of the same peptides that assemble together into this small little barrel. This is looking down the barrel. It's got a hydrophobic inside. It's kind of like the micelles I just showed you in the last handful of slides. It's got a hydrophobic core, hydrophilic outside when you put them in water. But they have this very specific shape. And so what does that do for assembly? That does the following. This is really what they look like in solution. And so if they're that perfect and they're that stable, you know exactly what every side chain is off of these amino acids. You know what all the functionality is, you know where it is. You can put what you want where you want because they're computationally designed in the Savant Group. They're not from nature. They're completely synthetic. So that's a really rich building block and something we're doing all kinds of things with. I just want to highlight a couple of foundational examples. One is this original work with Christy and with Jeff from Mike Hader's thesis, where Jeff designed the peptides to assemble into the building block and then designed the building block to assemble into a very specific lattice. You're looking down at the coiled coils here. This is a very specific symmetry. Point is, the molecule was designed to make just this exact symmetry, and in fact it did. When you put it in solution and then look at the particles formed and then analyze the nanostructure, it in fact is exactly this symmetry that was predicted. So you skip all the intuition, you skip all the experimentation, you make that one molecule, put it in solution, it makes exactly the nanostructure you predicted. So that really highlights Jeff's magic with respect to his computational prediction. What it also highlights is the fact that professors can be really slow about papers. So Mike graduated in 2018. Still a really nice paper in the works with Mike. He's got like a family and a job, and they're kind of busy. So maybe by the time I retire, that paper will come out. But it's going to be a nice paper. This is Yu's work. I showed you one that worked just now. A lot of them didn't work. They came close to working. So here's an example, another lattice symmetry, another peptide that was designed to make this. If you look at it in the TEM, you have some square-like particles and then a bunch of ugly particles. If you look at it in high resolution in the TEM, it's 20 nanometer scale bar. The nanostructure now is close, but a little off in spacing, so the symmetry is slightly different than what was predicted. So that feeds back into Jeff's theory for future predictions. But this is a material science department, and so UTN, this was his thesis work. He's not beholden to biological conditions. These aren't to put in the body. This is to make nanolattices. So he started changing solution conditions, like instead of just ambiently cooling the solution from high temperature, he did isothermal quenches, really controlled the nucleation and growth of things, and got really beautiful particles. So there's material science to be done with self-assembly, just like people control things like crystallization. He also changed the pH of these, didn't worry all about the lattice that was supposed to form, made these, discovered this really homogeneous nanotube phase. So again, your professor tells you to do something, go into the lab and just ignore them sometimes. You come back with some really amazing results that we didn't predict. And then to end, shout out to Chris Clarkson for the nomenclature here for these building blocks, which are now called bundlemers because of Chris. They're called bundlemers for the following reason, and that is, I just showed you some physical assemblies of these, but you can also covalently stick them together into chains. So you can consider them like a monomer, but it's a big monomer, right? It's a bunch of peptides collected together into this barrel. Chris is like, hey, we should call them monomers. We call them monomers all the time, but other people call them that now too. So that's really gratifying, and it makes sense. It makes sense scientifically as to why they're called monomers. So here's two examples, yellow and purple. Don't worry about the peptides, but they have two reactive ends on both ends of the bundles that want to connect them together into a chain, into a polymer chain. This is actually an image from our first proposal that we wrote to the Department of Energy, which we didn't get. The reviewers wrote back and said, hey, you guys made this cartoon like just a completely rigid rod. Like, you don't know that because we didn't have any data. We just had PowerPoint data. We were like, oh, that's a good point. It's probably going to be like a flexible polymer chain. But then when we got the data, we in fact saw just absolutely rigid polymer chains. This is a micron scale bar. The diameter of these things is 2 nanometers. So in the electron microscope, they're just stiff forever, which is just exotic. Really weird. Polymers don't like to be this stiff. Something we're still exploring as to why they're that stiff. Again, in the scattering, you can see individual building blocks characterize their length and their diameter. You can characterize the chains too, and it's just telling you, here's that -1 slope I showed you earlier. It's telling you you've got some one-dimensional thing, but it doesn't relay to you exactly how stiff they are. In fact, they're just amazingly stiff, something we're taking advantage of to make liquid crystals, to make fibers, to make films, something we're trying to get some new IP on to really take advantage of. But that stiffness is really exotic. Here it is in cryo-TEM, also showing it's not from just drying it out. And just to highlight how stiff it is, this is an example for those of you interested in polymer physical chemistry. Here's persistence length, or how stiff the polymer chain is characteristically relative to its mass per length. And here's it's a nice quadratic relationship of persistence length versus mass per length. So typical hydrocarbon polymers are way down here at angstroms to nanometers. DNA is tens of nanometers in persistence length, a little bit more massive. F-actin and microtubulin and viruses get a lot more massive and a lot more stiff. But ours, we estimate at this point, are somewhere around right there. But we think that's the lower limit. This is estimated from TEM. We think they're just a lot more stiff than that. So that's something we want to quantify going forward. And it's still a little bit mysterious, but that's why we're all here doing research to try and figure these things out. Okay, I want to end with a handful of thanks and gratitude. First and foremost is central facilities on campus allow me and anybody else to do these kind of things. These kind of tools are just absolutely critical to modern research. If they don't exist, the research doesn't get done, and that's the forefront of where things are. This is old equipment here. This was the initial microscope that we used. Chow's smiling a little bit because it was not the easiest situation to do cryo-TEM on. This is our newish microscope. It's now 12 years old, but still a workhorse over in the Keck lab. But most importantly, Chow, Yong, Yu Ying, more recently Yao Ping, Chow Ya helped, Townsend, and then importantly, people have left us in the past. Frank in particular, but also Jen Sloppy. Those people are why the lab works, because they're the ones that interact with the students to train them on the equipment to make them self-sufficient. And they still work, all the students still work with those people, right, every day when you're in the microscope. Really important. I'm highlighting microscopy because that's what I focused on, but all the other central facilities on campus too, just as critical for our research enterprise going forward. The staff in the MSE department, I want to highlight Susie, Lisa when she was in the department before she went up to the college, Kathy and Charlie, but more recently Brenda, Scott. Betty, thank you very much. This is such an old picture that don't tell her I said that. I didn't recognize her in that picture. I love Betty. Betty used to be in chemistry, then biology, so I met her back when she was in chemistry when I was working with Joel Schneider, as a matter of fact, so it was a long time ago. But nothing gets done without a really great group of people to work with every day. On the business end of things, I wanted to put one more slide with students because I forgot Ying Chow on that earlier slide. I noticed earlier today, I was like, oh my God, one more student that I forgot. This was during the 20th birthday party of the material science department over in the ice lab. Here's Tuna again, Matt, Hongong, and Balent. But one thing I wanted to highlight, oh, there's Chris and April hiding over there too, just celebrating the 20th birthday party. But I wanted to highlight this because one of the things I find most gratifying about working with colleagues and with students is the time you spend outside of the lab and outside of work, because then everybody realizes everybody else is a person and has a personality and has lots of fun things to say and lots of strong opinions about things sometimes. And these are the times when you discuss these things. This was a group dinner at the American Physical Society meeting in Chicago. This is when UTN had graduated and he was a postdoc at the University of Chicago, so he picked out a barbecue place. And we went and we had a really fun time for a handful of hours talking about all kinds of things. So those are really fun experiences. This is a more recent, more athletic endeavor. This fall at the Susquehanna, at a state park at the Susquehanna. With lovely hike, and I hope if you haven't been there, you should go. It's not far away, and it's just a beautiful hike along the banks, essentially the cliff banks of the Susquehanna. But during this hike, well, first of all, we almost lost Yao, so I'm glad we didn't lose him. He's here, so he survived. But you also, you get a glimpse of what, like I said, people's personality and what they're really like. And you get people out of the lab, they kick back and relax in really weird ways. So this is what a bunch of my group decided to do, and just relax and take a break during the hike. I think it's a really good album cover or something, but I'm definitely going to use this again in the future. Don't tell the safety people. Importantly, I want to give a shout out to my dad and my mom, Janet and John. This is my mother. This is her birthday. Fig's trying to help. We lost Fig recently. Mom's fine. A lot of you have met Fig over at our house over the years, but 18 years he made it. So he had a good run. Mom's doing great, and my dad here is doing one of his favorite things. This is his 82nd birthday a handful of weeks ago. He busted out a 96 single varietal Cabernet Franc from Foxen Vineyard, if you're familiar, north of Santa Barbara. Great place. That wine was really good. So that was a good one to celebrate with. I want to highlight my kids, because they've been along for this entire ride. This is 18 years ago. This is Alec, that's Hugo, and that's Max. They're about 3-ish, he's about 5-ish. That's on the back deck. They don't look like that anymore. They look like this now. This is at Max's college graduation just last week. This is Alec, Max, and Hugo. This is Max hanging out with his buddies, his housemates this last year. Just so much hair. It was so much hair. It was just everywhere. It was just hair just everywhere. Clearly the mom's genes, not the clearly the dad's genes. It was so much hair, but I wanted to point out Max, he knows it, but he is the namesake of the Max series of peptides. So not many of us can say we've got a peptide named after us forever in the literature, but Max can. And then finally, I wanna say something about my wife Beth. So when we first met, she was in law enforcement. So this is her. She was in the Charlie's Angels for a handful of years. For those of you under 30, you might not know what the heck I'm talking about, but she was a Charlie's Angel, but then she decided law enforcement wasn't for her, and she went to graduate school. And when she went to graduate school, she succeeded. Now she's succeeded in her career. She won a really big national award from the American Psychological Association last year. That we went to DC and had a really fun time during her symposium at the conference. So very accomplished. We also found that she's got really cool scientific lineage in her family. So we looked, and then we looked it up, and she's got an uncanny resemblance to Charles Darwin. It's just amazing. So you think you know somebody your whole life, and then something like that happens. But Beth, in all seriousness, 30 years and saying things like thank you is just way too trite. But thank you for being with me, being for me that entire 30 years, and looking forward to 100 more. Thanks a lot.
Okay, I think we have time for questions and maybe some answers.
Maybe.
Maybe I'll ask a quick question. I mean, not
Sure.
being in this area. You're talking about the stiffness.
Yes.
You had the length, like a length scale versus a mass.
Persistence length versus linear mass density. So how massive per
So
length.
can you measure can you measure some physical properties like with nano indentation or something like that to really get something that's more,
Sure.
maybe it is a real strength, but it just, if you're a mechanical engineer, I think you think of pulling it or trying to.
Yes, yes, and yes. So the question is, can you physically measure the stiffness of these nano rods? And the short answer is yes. It's just really difficult to figure out the right way to do it. So we estimated those stiffnesses just based on tracing the trajectory of the chains in microscopy, which is not a great way to do it, but that's what we had at the time and have at the time, and it's easy to do. But the thing is, if it's completely stiff for a micron, like it's a little meaningless to start projecting how stiff it is beyond that, right? Because you want to see it bend at some point, then you can back out how persistent it is in its trajectory. So you would want to connect it to something, pull on it, bend it to see exactly how stiff it is. That's something we're going to try and do with particles. So I'm pointing at Eric right now. Maybe I'm going to point at Kenny too. Connect them to colloidal particles. Then you could do laser tweezer experiments. That's a great way to get at it. You could maybe do AFM, but that's a really challenging measurement. There's lots of ways we've been thinking about doing it. What I'd like to do is to grow them long enough, many hundreds of microns, where then we could just label them and do fluorescence microscopy and just watch them fluctuate. That's the way they quantitated a lot of those persistence lengths of things like F-actin, bacteriophages, viruses, DNA, because you can make them nice and long, you can just see them. Back to the microscopy being important, you can just see them fluctuate around. But all those are possible, but the measurements are a challenge. Jill.
Thanks for an impressive and fun presentation.
Oh, and I should have thanked you. Yeah, I'm so anxious to. Go ahead, Joe.
It seems like a lot of your work's experimental in nature. You did hint a little bit at some of the computational work. I suspect that is underlying a lot of your structures. I guess I'm wondering, what's the potential of computational work at maybe predicting some of these self-assemblies? And actually, I was wondering about AI being able potentially to drive to see which ones are going to be most valuable.
Yeah, so great points, Jill. So a couple things. One is, I'll just focus on the end part with the bundlemers. So we've got a growing library of peptides now that are designed to make the coiled coil particles, right? That then we're using to build things with, connecting them together, assembling them together. So we're building that library that I think at some point kind of soon we will start to have a data set with which one could start training a predictive code to come up with new sequences for desired coiled coil structure. The thing is about coiled coils is there's already really good heuristics as to what it takes in a molecule to form it, right? So it's not that exotic to actually form the coiled coil. What's exotic is to dial in any kind of random chemistry you want. So, and non-natural functionalities too. So that's where machine learning might become important once our data sets gets bigger and bigger and we can start using that as a training set to design new molecules. Another part important for computation is, and this is actually going to be really fun research, I didn't show any of the results today, but a lot of the students now are making the molecules, making the coiled coils, and then putting them in solution. But we're making them, for example, sometimes they only have one charge type. So they're not mixed, like proteins have positive and negative charges. We can make them with just one charge type. We can make a mixed charge sequence that has the same charge, right? So it can have two bundles, same charge. They should act exactly the same way as little nanocolloidal particles. But you put them in solution, the one makes these just beautiful liquid crystal phases, and the other one just makes this amorphous kind of mess. That's not, I would not have hypothesized that. Like that's just kind of weird. And so now it's those kind of things we're feeding into ideas for simulations, right? Simulating, hey, how are those particles interacting for the one to make this beautiful liquid crystal phase, and why is this other one just making this amorphous junk? If you had done the simulation first, you would have got a result, right? And we're doing that, and sometimes they match experiments, sometimes they don't. So that's going to be important vein of research for a long time to try and close the gap between the in this case, molecular dynamic simulations and the actual experimental results. So I think it's really rich stuff to be done in there. Study patchiness of charges. That's something we're really excited about, a bunch of us, collaborative work. How about Beth? Got any questions? She hears about
About
this stuff. all
kinetics. So you talked about some of these being kind of locked in a kinetic, I think I heard you say sort of a kinetic well.
Yeah.
So how do you know? I mean, I'm a catalysis person, so you know temperature and pressure, all of these other things. The conditions that you're using, if you tried a slightly different set of conditions, I mean, maybe you do this. Can you get totally different structures, and can you predict that in any way?
So focusing on the last part with the bundlers, the interesting thing with those, with respect to kinetics and stability, is that the designs, usually for the best, sometimes it's difficult, the designs are made so that the particles are really stable, okay? So any kind of solution conditions we use, we first and foremost want to make sure the particles are intact, because then we can study how they interact, like we were just talking about, to build up superstructures to connect them together. And so then, in those processes, we study different temperatures, pHs, salt, right? How can you control how they interact to assemble into these bigger nanostructures, or do covalent chemistry to link them together? So that's where we explore lots of different kinetic effects on the polymerizations, on the assemblies. Can you anneal them? I showed you one example of the doing an ambient cooling versus an isothermal quench, where you get this kind of a mess, but then all regular particles. So absolutely. So all those tools work if one's cognizant of whether the building block is stable or not. If it's not stable and it started to unfold during those studies, then it would be a complicated mess if you had individual peptides and not just the folded monomer particle. But yeah, all that's important for sure.
There you go. What was your name?
Kenny.
Kenny, okay.
So, great, awesome, Darrin. Learned a lot, I think. So, maybe this is a little softball of a question, but you're showing, and you started to talk with these beautiful structures that nature's developed that have all these designs and structures, and I sort of know the answer to this, but I want to hear what your thoughts in terms of designing the three-dimensional structures, or things with design beyond just the solution assembly of the bundlers, or the one-dimensional structures.
Yeah, I don't think that. So the question is, we talked a lot about designing the particle and then designing kind of one hierarchy up with respect to interparticle structure, but can you go beyond that, another levels of hierarchy? And I don't think that's a softball question at all. That's the viruses are a great inspiration for that, right? So at this point, as you know, we design the peptides to make the particle, and then we've got our toolbox of our particles, and can we mix and match those or put functionality on them to make them zip up into some higher order nanostructure? But then so far, we just have experimental discovery of any higher order effects. So sometimes we see things chain into one-dimensional things, but then they make beautiful liquid crystal phases. So they interact with each other to make a third order of hierarchy structure. Other times they'll be designed to assemble into a sheet, but then the sheet will curl up into a tube. Those kind of higher order effects we haven't purposely designed in yet. But that's the end target for self-assembly, right? So you can make a spherical micelle anytime you want, which is great because all the fundamental research people have done, people know how to do that. You can do that. Anybody can do that. But can you make the particle, but then have the particle interact with other particles to make X? That's the forefront right now, and I think we're right there with the team of people doing all of these things. But that level, but then a level above it, yeah, that's where everything's headed.
Well, there are no additional questions, let's thank Darrin again for a wonderful.